Our proposed idea is about simultaneous scoring of clusters in recursive cluster elimination, which is applied on transcriptomic data analysis. Our motivation is that gene expression data analysis is a challenging task due to the small sample size and high dimensionality of the data, and the feature selection algorithms is a useful approach to deal with high dimensionality. SVM-RCE, which stands for Support Vector Machines in Recursive Cluster Elimination, is one of the few techniques created for feature selection in high dimensionality data. An SVM-RCE approach has been utilized for detecting distinct clusters of genes that exhibit varying levels of expression in the presence of pathological states. So in the original approach, the genes were grouped into clusters utilizing the K-means algorithm. For each cluster, a corresponding subdata is extracted, where this subdata contains gene expression values just for those selected genes, retaining the original class levels of the samples. Then a score was assigned to each of these clusters by running support vector machines and by applying internal cross-validation. Following that, a percentage, which is mostly 10% of the clusters with low scores, are eliminated. The elimination is performed by filtering out of the genes that are members of those lower score clusters. The mentioned procedure is iteratively executed until a specific number of the cluster persists. The present study proposes a novel approach for scoring the clusters simultaneously by applying linear support vector machine or random forest on the cluster centers. The absolute values of the coefficients given by the linear support vector machines serve as the score of the corresponding cluster. Similarly, the feature weights given by random forest serve as scores of each corresponding cluster. In our experiment, we use 17 different datasets obtained from Gene Expression Omnibus database. The results indicate that the performance of the new approach is comparable to that of the original approach. We can see that different classification and model performance in the terms under the curve, number of the genes, and execution times. Number of the genes is represent the remaining number of the genes from each iteration, after each iteration. We can see that the blue ones is the under the curve values, and under the curve values is the similar to each other. In the left side, it's the original approach, and the first two graphics shows that our new approach results. And the blue box shows that or under the curve performance. So performance is comparable with original one. And but original one have more remaining genes in the different from the new approaches. But in here, our most important values is that execution times. So in the original approach, the execution times take 21 seconds, but our new approach only needs 4 seconds. So now or new approach is faster, 80% faster than the original approach. So in this technique, we will facilitate disease diagnosis in addition to enchantment or comprehension of the molecular mechanisms underlying disease onsets and the progressions.